So if you take like a prize-winning sea-level dairy cow and just drop it in the Himalayas.
I was going to have a terrible time.
Right, exactly. I mean, its cells will immediately start screaming in this state of biological panic.
Yeah.
Every microscopic system inside its body goes to war just to keep the animal breathing.
Yeah, but then you look at the native Ladakhi cow standing right next to it, and its cells are practically asleep.
Which is wild, and that's exactly what we're getting into today. We are looking at the literal genetic blueprints of survival. Welcome to today's deep dive, where we're unpacking a really fascinating research article titled Overexpression of Genes Associated with Hypoxia in Cattle Adapted to Trans-Himalayan Region of Ladakh.
Quite a title.
Yeah, a bit of a mouthful. But our mission for you today is to uncover the hidden, basically the mechanical secrets of how life survives in some of the most punishing environments on Earth. And, what a cow's molecular toolkit can actually tell us about biological efficiency.
It's honestly a phenomenal case study in evolutionary engineering. I mean, when we discuss the Trans-Himalayan region, specifically Ladakh, which is often called the land of high passes in India, we're dealing with an ecosystem that actively degrades the physiological functions of the organisms living there.
Right. It's not a friendly place to live.
Not at all. You're looking at elevations above 3,000 meters. Winter temperatures can plummet to like minus 40 degrees Celsius. You've got intense ultraviolet radiation, humidity hovering at a bone-dry 25 to 40%, and barely any vegetation.
Just a brutal setup.
Yeah. But the absolute most critical threat to survival up there is the hypobaric hypoxia.
Right, hypobaric hypoxia, meaning the air is just impossibly thin. Up there, the oxygen availability is only, 60 to 70% of what you and I are breathing right now?
Exactly.
So for a massive 400-kilogram ruminant, just maintaining normal bodily function isn't just difficult. It requires a complete metabolic overhaul.
It really does.
And I think it makes you wonder, why are scientists so hyper-focused on a cow on a mountain?
Well, the implications go way beyond agriculture. Because if we can map exactly how a mammal adapts to chronic oxygen deprivation, we unlock data that is critical for biodiversity conservation, veterinary medicine, and even human biomedical research.
Like studying diseases involving hypoxia.
Yeah, exactly. Conditions like ischemia or severe respiratory failures. Plus, we really have to consider global climate resilience.
Oh, for sure. Because environments are shifting so rapidly right now.
Right. And understanding how livestock maintain productivity under extreme physiological stress is crucial for global food security. And these native Ladakhi cattle are walking masterclasses in this. I mean, they subsist on terrible quality feed, endure extreme water scarcity, breathe that incredibly thin air, and yet...
They still produce milk, right?
Yes, 3 to 4 kilograms of milk a day. They serve as the absolute nutritional lifeline for the local communities up there.
So to figure out how they actually do this, the researchers had to look under the hood. And to really understand the mechanics of this study, we first need to look at how hypoxia fundamentally assaults the body's cellular engine.
Yeah, that's a good way to frame it.
Because if we view oxygen as the essential fuel required for the mitochondria to generate ATP, which is the energy currency of the cell, hypoxia is essentially a severe fuel line restriction. It's like trying to drive a car up a steep mountain with a clogged fuel line.
That's a great analogy, but I'd refine it just a little bit. It's not just that the engine sputters and slows down. The cellular engine doesn't just passively fail.
Oh, really?
No. The moment that fuel line restricts, the cell actively mounts a massive coordinated defense. It completely alters how it produces energy, basically attempting to prevent a total biological shutdown.
Wow. Okay.
And it does this through targeted gene expression.
Yeah.
I mean, every single cell in that cow's body contains the entire genetic blueprint for survival, right?
Right. Thousands of genes.
But those specific survival genes are dormant under normal sea level conditions. They only get transcribed or switched on when the cell detects an environmental crisis.
Which brings up a pretty big logistical problem for the scientists. Because if I'm a researcher trying to understand this invisible genetic defense system, I can't just, cut a living cow open to look at its lung or heart tissue to see if the cells are suffocating.
No, definitely not.
So they must be using blood markers, like analyzing the exhaust to figure out how the engine is running deep inside the machine.
That is exactly how they did it. The researchers isolated peripheral blood mononuclear cells, or PBMCs, from healthy heifers.
Wait, heifers? Just young cows?
Yeah, young female cows. So they pulled blood from native Ladakhi cattle, Holstein Friesian crossbreds, and exotic Jerseys that were living in the high-altitude mountains. And then they compared them against Sahiwal, Holstein Friesian, and Karen Fry's cattle living down at sea level in the tropics.
Okay, so a clear high altitude versus low altitude comparison. But why specifically those PBMCs from the blood?
Because PBMCs are a highly reliable, non-invasive proxy. Blood is constantly circulating through every single oxygen-deprived organ in the body. So the gene expression profile of those immune cells in the blood accurately mirrors the systemic physiological stress that the entire animal is experiencing.
Okay, that makes perfect sense. And let's unpack the extraction process, because since they're looking at gene expression, the researchers extracted RNA, not DNA.
Right. They completely bypassed the permanent DNA blueprint and went straight for the active transcription work orders, the mRNA.
Right, because if DNA is the master blueprint in the office, RNA is the actual work order being sent down to the factory floor.
Exactly.
So to measure those work orders, they synthesize complementary DNA from the RNA and use this technique called quantitative real-time PCR, or qPCR.
Which is just an incredibly sophisticated mechanism for molecular quantification. Rather than just looking at a sample under a microscope and guessing, qPCR uses thermal cycling to exponentially amplify specific target sequences of the DNA.
Like a molecular magnifying glass.
Exactly like that. By attaching fluorescent probes that light up during the amplification process, researchers can precisely quantify the initial amount of mRNA present.
That's amazing.
Yeah, it tells us not just if a gene is turned on, but exactly how loud that gene is screaming.
I love that. So when they measured that fluorescence, they didn't just find a random assortment of active genes. They found a highly specific tiered survival strategy. And it all cascades from one central regulator, right?
Right. Hypoxia-inducible factor 1, or HIF1.
HIF1. Okay, so the researchers found a 2.52-fold increase of HIF1 alpha in the high-altitude cattle. And from reading the study, I basically look at HIF1 as the ultimate biological tripwire.
It functions exactly like a tripwire. It's the master oxygen sensor.
Mm.
Under normal oxygen conditions, the HIF1 alpha protein is constantly being synthesized and then immediately degraded by the cell. It's just a continuous rapid cycle. But the moment cellular oxygen drops, the degradation process stops. The HIF1 alpha stabilizes, accumulates, moves into the nucleus, and physically binds to the DNA to trigger the transcription of hundreds of downstream survival genes.
It's basically the emergency control center sounding the alarm.
Yes. And alongside another transcription factor called EPAS1, which, by the way, is very famous for its role in the high-altitude adaptation of Tibetan human populations, HIF1 initiates a complete biological overhaul.
Okay, let's break down that overhaul for the listener, because the study doesn't just list off random genes. It reveals two massive coordinated strategies the cell uses to bypass that clogged fuel line we talked about.
Right.
And the first strategy is a complete vascular reconstruction, building new highways. The tripwire triggers genes like VDGFA, NOS2, and ECE1.
And the numbers on VGFA are wild. They saw a massive 6.88-fold increase.
Nearly 7 times the normal amount.
Yeah. This gene physically signals endothelial cells to divide and build brand new blood vessels, which is a process called angiogenesis. Basically, if the existing highways can't deliver enough oxygen, the body just aggressively builds new ones.
But building new vessels takes time, though. Like, the body needs an immediate fix to run alongside that long-term construction project.
That is exactly where NOS2 comes into play. NOS2 produces nitric oxide, which is a highly reactive gas. When nitric oxide diffuses into the smooth muscle cells surrounding your existing blood vessels, it forces those muscles to relax.
Oh, so the blood vessels literally physically dilate. They get wider.
Yes, exactly, to let more blood flow through instantly.
But wait, if NOS2 is drastically widening the existing vascular highways to let more blood through, wouldn't that drop the animal's blood pressure to, like, dangerously low levels? I mean, an animal already undergoing severe physiological stress, plummeting blood pressure seems like a fatal flaw.
It would be if it weren't so localized and tightly regulated. The beauty of this system is that the vasodilation is targeted specifically to tissues that are screaming for oxygen, while other systemic regulators like ECE1 help manage overall vascular function to prevent total cardiovascular collapse.
Ah, okay, so it's very targeted.
Very. Plus, you have inflammatory signaling molecules like TNF-alpha being upregulated due to the tissue damage caused by the hypoxia. And you've got the glucocorticoid receptor alpha, GR-alpha, ramping up to help the body manage the deep systemic physiological stress of the entire ordeal.
So it's a very delicate, high-stakes balancing act to keep the blood flowing without just completely crashing the system.
Exactly.
So strategy one is forcing whatever little oxygen you have into the tissues by building new pipes and widening the old ones. Strategy two is entirely metabolic, and this is where the numbers and the data just completely blew my mind.
Oh, the glucose transporter data.
Yes. They looked at a gene called GLUT1, which is a glucose transporter. In the high-altitude cattle, they saw a staggering 18.32-fold increase in GLUT1 expression 18 times.
It's massive.
The cells are in a total oxygen shortage, so they are literally panic buying sugar.
Yeah, you have to look at the mechanics of cellular respiration to understand why they are pulling in so much glucose. Normally, a cell uses oxygen in the mitochondria to break down glucose incredibly efficiently. It's a process called oxidative phosphorylation, and it yields a massive amount of ATP.
The normal fuel process.
Right. But without oxygen, the mitochondria just can't run. So the cell is forced to shift to a secondary emergency pathway called anaerobic glycolysis.
Which happens outside the mitochondria and doesn't require oxygen. But the catch is that glycolysis is horribly inefficient, right? It produces just a tiny fraction of the energy compared to the oxidative pathway.
Precisely. To generate the exact same amount of energy just to keep the cell alive, the engine suddenly requires massively more fuel. Hence the 18-fold increase in those GLUT1 transporters. They are actively dragging every molecule of glucose they can find from the blood into the cell.
Just hoarding it.
Yeah. And alongside GLUT1, the researchers found a massive upregulation of HK2, which is the enzyme that actually initiates the metabolism. It traps that glucose inside the cell and kicks off the glycolysis process.
So the cell is desperately hoarding sugar and burning through it terribly inefficiently just to keep the lights on while that oxygen tripwire blares in the background. It is an incredible, violent, microscopic war.
It really is.
But here is the most brilliant twist in the entire research paper. Because up until now, we've just been comparing the cattle in the clouds against the cattle on the beach, basically. But the researchers didn't stop there.
No, they didn't.
They compared the different broods within the high-altitude group.
Right. They analyzed the native Ladakhi cattle, who have naturally adapted to this region for centuries, standing side by side in the exact same thin air as the exotic tourist breeds, like the Holstein Friesian crosses and the Jerseys, who were imported to the mountains relatively recently.
And logically, your brain assumes that the native Ladakhi cattle, the absolute masters of this environment, would show the highest expression of these hypoxia survival genes. You'd think their vascular construction crews and the glucose panic buying would be off the charts compared to the others.
That's what you'd expect.
Right.
But the data showed the exact opposite.
The exact opposite. When compared directly to the tourist Holsteins and Jerseys living in the exact same environment, the locally adapted Ladakhi cattle showed significantly lower mRNA expression of that master switch HIF1 alpha. They had lower VGFA, lower ECE1, and drastically lower expression of that glucose transporter GLUT1.
It is a profound biological paradox, at least until you reconsider what gene expression actually represents.
Right.
The high mRNA transcription levels in the exotic tourist breeds aren't a sign of mastery. They are a sign of severe biological panic. Those tourist cows are not biologically adapted to the hypobaric hypoxia. Their tissues are literally suffocating.
So their HIF1 tripwires are just constantly firing.
Exactly. They are desperately upregulating their genes, forcing their cellular engines to redline in first gear just to barely survive the day.
But the native Ladakhi cattle have naturally evolved a fifth gear. Their baselines are entirely different. Over centuries of generational selection, their actual physiology has structurally adapted to the environment.
Right. Maybe they have naturally larger lung capacities, or hemoglobin with a naturally higher affinity for binding oxygen. The study doesn't measure their physical organs, but the low gene expression proves that their actual tissues simply aren't experiencing the same level of cellular starvation.
The Ladakhi cells don't need to panic by glucose or sound the HIF1 alarm because, from their cellular perspective, there is no crisis.
Exactly. Their cellular baseline is already optimized for a low oxygen environment. It really demonstrates a fundamental rule of evolutionary biology. True adaptation isn't about working harder to fight the environment. It's about altering your biology so you don't have to fight at all. The Holsteins are surviving desperately, but the Ladakhi cattle are surviving efficiently.
Wow. And the timeline of that adaptation is what really sticks with me. We are looking at a deep genetic calmness that took centuries, if not millennia, of harsh culling and natural selection to lock into the Ladakhi germplasm. The tourist cows simply haven't had the evolutionary time to shift their baseline.
They haven't.
Oh, and speaking of shifting baselines, I cannot let us finish without mentioning the heat shock protein plot twist.
Oh, I love this part.
It's so good because it perfectly illustrates how incredibly sensitive this genetic machinery is to localized, immediate threats.
Yes. So the researchers also measured heat shock proteins, specifically HSP27, HSP70, and HSP90. These are essentially molecular chaperones. When a cell experiences severe stress, particularly from extreme temperatures, its internal proteins can start to unfold and denature.
They kind of melt.
Yeah, in a way. And heat shock proteins rush in to physically stabilize and fold those proteins back into their correct functional shapes, which prevents cellular death.
So you would naturally assume that the cattle living in the brutal, freezing, high-altitude extremes of the Himalayas would have massive levels of these stress proteins. I mean, they're on a frozen mountain.
Right. But once again, the data completely flipped the script. The expression of these heat shock proteins was actually significantly lower in the high-altitude cattle and massively upregulated in the tropical sea level cattle down in the lowlands.
Which seems totally counterintuitive until you factor in the specific timing of the methodology.
Exactly. The samples in Ladakh were taken during the month of August. High up in the mountains, August provides a very comfortable ambient temperature of roughly 25 to 30 degrees Celsius. It is practically a vacation for a cow up there in August.
So the hypobaric hypoxia was stressing them out, but the temperature was totally fine.
Exactly. But those low-altitude tropical cattle, the Sahiwal and Karen fries down at sea level, they were also sampled in August on the Indian plains. They were suffering through massive, sweltering summer heat waves and severe humidity.
Oh, man.
Yeah, so their oxygen levels were perfect,
Yeah.
but the intense thermal stress was causing their cellular proteins to denature.
So their cells were desperately churning out heat shock proteins to survive the summer weather.
Yep. It is a brilliant reminder that an animal's gene expression is a highly localized, real-time reflection of its immediate environment. The Himalayan cattle were fighting suffocation, while the tropical cattle were secretly fighting hypothermia.
You really do have to check the local weather report when you're reading a molecular genetics paper. That is just fascinating. So to pull all of this together for you listening, we started by looking at the incredibly punishing environment of the Trans-Himalayas and that hypobaric hypoxia that threatens every living thing up there.
Right.
We used the circulating blood cells as kind of an exhaust proxy, observing how the master tripwire HIF1 alters the entire blueprint of the body, triggering massive vascular construction projects and inefficient, panic-driven glucose metabolism. But most importantly, we discovered that true evolutionary adaptation, like what we see in those native Ladakhi cattle, isn't about sounding louder biological alarms. It's about naturally developing an engine so perfectly suited to the terrain that the alarms never need to ring in the first place.
Which really leaves us with a critical biological reality to consider. I mean, we've seen how centuries of generational exposure to severe extremes slowly crafted this genetically efficient, low-panic baseline in Ladakhi cattle. But the global climate is currently shifting at a pace that far exceeds the standard speed of natural evolution.
Oh, for sure. The timeline is totally compressed now.
Right. Heat waves, droughts, and shifting agricultural zones are fundamentally altering the environments our global food sources rely on. As environments change faster than natural selection can comfortably adapt to, you have to wonder how many of our agricultural species are currently running their cellular engines in that state of invisible biological panic.
Because we rely so heavily on specialized, highly bred animals like the Holsteins and Jerseys for global food production.
Exactly. And if we drop them into increasingly stressful changing climates, how long can their cellular engines run on emergency fuel lines and biological redlining before the systems begin to systematically fail? The Ladakhi cattle hold the genetic blueprints for resilience, but the question is whether we have the time to integrate those lessons on a global scale.
It is a profound realization that kind of changes how you view the intersection of genetics and the changing world outside your window. The survival of tomorrow might entirely depend on the invisible baselines being tested today.
Well, thank you so much for joining us on this deep dive. Keep asking questions, keep exploring, and never forget to marvel at the microscopic engineering working tirelessly inside your own cells. See you next time.